nice and easy. You'll get your results today. It's one of the few that we don't have to set up ahead of time. We just get positive and negative results. We are in this lab looking at two enzymes, catalase and oxidase. This is lab 24 as well. Catalase is an enzyme that some bacteria can make to degrade, this is hydrogen peroxide, H2O2. So bacteria, or at least some bacteria, can make it to degrade hydrogen peroxide. That's the stuff in the brown bottle that you pour on cuts, right, to sterilize them. If bacteria have catalase and they're using it to degrade hydrogen peroxide, they're degrading it into things that don't hurt them, harmless substances to them. Whereas down here at the bottom, my little note with the stars by it, you can see it. There you go. Peroxidase, this is the human enzyme for degrading hydrogen peroxide. This is what we use to break down the stuff in the brown bottle from the pharmacy. And if we break it down with our peroxidase, we're turning it into stuff that kills bacteria. Toxic oxygen-free radicals, things that will hurt them. So we got a little bit of an arms race here. They get to it first, they turn it basically into water. If we get to it, we turn it into free radicals that are designed to kill them. Now, not all bacteria can make catalase, right? Just like every other test we've done. Some bacteria are going to be positive, some are going to be negative. That's what we're trying to find out. Do they produce catalase or not? So this is a real easy test to do. Here you get a glass microscope slide. You're not looking at it under the microscope, you're just using the glass as the surface to perform the test on. And then you'll read lab 24 to see which two bacteria you're supposed to use for the catalase test. Your materials page is probably going to have like 4 bacteria listed, so read through the procedure, make sure you're using the right two for catalase, and just dry smear them onto the glass, 2 piles, whichever 2 bacteria your lab book says to use. Then get a dropper full of hydrogen peroxide. We've got the brown bottle from the pharmacy up here on the front table, some disposable pipettes. And you're just going to add a drop of peroxide to each pile of bacteria. Yes, ma'am?
What did you say, the positive?
I have it, yeah. But it's right there.
Well, no, that's the physical, right?
Right, that's what it looks like. What we're trying to find out is, do they make catalase or not?
Do it make, does it produce catalase?
So you're going to add peroxide, just drop it on with the dropper, and if you see bubbles, that means that they do have catalase, that they are able to degrade peroxide. And if they don't bubble, then they don't have catalase, they're not able to degrade hydrogen peroxide.
Are you calling the hydrogen peroxide our reagent?
Technically for this test, yeah. It's the only way you're going to know if they make catalase or not.
What was the positive you had catalase means?
Can degrade peroxide. Yeah. Positive looks like bubbles, positive means produces catalase. So negative looks like no bubbles, negative means does not produce catalase. So why do you bubble when you pour peroxide on a cut? You produce peroxidase. Right? That's why you bubble, from peroxidase. That's why I asked that question. Okay. So that is in your photographic atlas. I have the page written down. Look. 63. Catalase is on page 63 of your photographic atlas. Now your stock cultures today are on petri dishes because you need a lot of bacteria, so we gave you plenty to scrape up. We've got 3 petri dishes of each bacteria, so you're sharing this for your whole table. So if you get the plate first, don't scrape up everything that's here. Save some for the other group. Also, these have been in the incubator for longer than they should have since we had class canceled, and they're very, very stinky, particularly the green one. Pseudomonas aeruginosa is one of the smelliest bacteria that we work with. It smells like trash, old trash that's been sitting in the trash bag forever. So be ready to go and have this open for as little time as possible. It's not going to make you sick, it just smells really bad. So, fair warning. All right, so that's catalase. Any questions on that one? Okay, still in lab 24, the second enzyme that we're going to look at is oxidase. This particular enzyme is present in bacteria that have cytochrome C. Okay, so So if they produce oxidase, if they make this enzyme, that means that they have this protein, cytochrome C. And cytochrome C is a member of the electron transport chain. Remember that from lecture? Electrons fall down, protons get pumped out. Cytochrome C is one of those proteins that the electrons are falling down, getting passed from one member of the electron transport chain to the other. This is one of the members of the electron transport chain. So again, if bacteria produce oxidase, that means that they also have this molecule, cytochrome C, which means they have an electron transport chain. All right, so this time you're doing it on filter paper, which I know looks just like my picture of the last slide, so I labeled it for you. Filter paper. It's in a box up here on the front table. It's a big roll. Just pull yourself off a piece about maybe 2 inches long. You can just tear it. It's fine. For this particular test, you have to add the reagent first to the filter paper. And it's in ampules, like your Vokes Proskauer A and B, where you got to break the glass inside. This is in the box up here on the front table. Oxidase reagent, and you're going to make 2 wet spots. Give yourself 2 nice wet areas on the filter paper because you're testing 2 bacteria again. And then you're going to add bacteria. And if you read through the procedure for this, your lab book says not to use the inoculating loop because the metal is supposed to give you a false positive. I've honestly never had a false positive, so you can use the loop if you want to, but if you want to follow the rules of your lab book, there's also some wooden sticks, applicators on your table in a box. Allegedly, the metal gives a false positive, but I've never seen it, so it's up to you. Again, whatever you decide to use, you're going to be scraping up a lot of growth from the Petri dish and smearing it into the wet filter paper. Try not to get too much agar. Try to get the bacteria off the surface. Don't dig into the agar and smear it on. And if the bacteria produce oxidase, okay, then that little wet spot's going to turn blue. Almost like your Simmons citrate, a really bright blue color the paper's going to take on. And if they don't produce oxidase, then you're not going to have any color change. It's still going to look like wet paper. So no blue color. And that one's on page 87 of your photographic atlas. Oxidase is on page 87. Questions on the oxidase test? Alright, so that's nice and easy. Lab 24, you get your results today. There's no incubation. Lab 25, we're back to our, have to inoculate tubes, put them in the incubator, come back on Monday and get the results. The name of the lab for lab 25 is nitrate respiration. We're actually going to be looking at 2 separate or related processes. This is part of the nitrogen cycle. Most of the nitrogen on the planet's surface, at least, is nitrogen gas. Most of our atmosphere is nitrogen gas. We've got more nitrogen than we do oxygen in what we breathe in. But we can't use nitrogen directly from its gas form. Neither can most bacteria. So there's a whole cycle for getting that gas form of nitrogen into usable forms, and this is part of that process. You okay?
Yes.
I thought you had a question. All right. So, nitrate, obviously a nitrogen-containing compound. NO3 minus, it's a negative charge there for the formula for nitrate. Some bacteria are capable of this arrow, nitrate respiration, where they convert nitrate into nitrite, NO2 minus. So nitrate respiration is the conversion of nitrate into nitrite. And some bacteria are capable of it, and some aren't, just like every other test we've done. Then some bacteria, after doing nitrate respiration, after converting nitrate into nitrite, can all go through a second process called denitrification. These are all bold words in your lab book here as well. Denitrification. And that is the process of converting nitrite into nitrogen gas and nitrous oxide. So denitrification converts nitrite into nitrogen gas and nitrous oxide. Some bacteria can do only nitrate respiration, convert nitrate into nitrite, and then they stop right here. And they're not capable of denitrification at all. Some bacteria can do both, and nitrate respiration and denitrification and end up all the way over here. And then there are some bacteria that can't do either one. So we've got three possibilities here. Bacteria can do nitrate respiration but not denitrification. Bacteria can do both, or bacteria can do neither.
And the last, the end one right there, that's nitric oxide, nitrous
Nitrous,
oxide.
yeah, Nitrous, nitrous oxide. And nitrogen, that little G is gas. So again, three possibilities. Positive for nitrate respiration, but negative for denitrification. Positive for both, negative for both. So how we're going to test which of those three options our bacteria are. We're going to be using nitrate broth, which, what do you think it has in it? It's nitrate, right? We're giving them a source of nitrate. And it's also got that Durham tube for collecting gas. What kind of gas are we hoping to collect this time? Nitrogen gas, right? Not carbon dioxide like with fermentation. Okay? That's why that little G is there. This is our gas, nitrogen gas. All right, so today it's pretty easy. You're just inoculating the tubes. And Lab 25 has a bunch of bacteria listed. They're on Petri dishes here. Okay, so you're just going to be using your loop. This is a liquid media, so you can just use your loop, put the bacteria in. The only other difference for inoculating is that you've got one of your materials listed is to use a soil sample. Because a lot of the bacteria that live in dirt can do these processes. So we've got a little Ziploc container here of dirt, but keep it fresh. Just take a scoop of the dirt and put it into one tube. Obviously, you don't have to sterilize the scoop or the lid, it's dirt. Right? So just one tube of nitrate broth gets a little bit of dirt from that container, and that's your source of bacteria for that tube. The rest of them, it's from the stock cultures and Petri dishes that are listed in Lab 25. So you put them in the incubator, that's all you do today. When we come back on Monday, then we have to add reagents to find our positive from negative. So we've got a two-step process here. Every tube, we're going to add nitrate A and nitrate B. It's another two-part reagent like your VPA and VPB. Okay, so every single tube gets both chemicals, nitrate A and nitrate B. Those two chemicals will turn red if there's nitrite present in the tube. Okay, so the chemicals nitrate A and nitrate B change color, they turn red in the presence of nitrite. So if the tube turns red immediately after adding nitrate A and nitrate B, it's because there's nitrite in the tube, right? Because those two chemicals turn red in the presence of this stuff, which tells us that the bacteria is positive for this first arrow of nitrate respiration. They made nitrite, and they stopped right there. Because if they had gone through denitrification, would there be any nitrite left in the tube? No, they wouldn't have turned red. So, positive for nitrate respiration and negative for denitrification if the tube turns red immediately. Add nitrate A and B, turns red, positive for nitrate respiration, negative for denitrification. They stopped right here. Now, if the tube doesn't turn red after adding nitrate A and nitrate B, there could be two reasons for that, right? The bacteria could have stayed right here and did absolutely nothing, didn't do either one, or the bacteria could have gone all the way down to here and doing both processes. We need to figure out which one it is. Did they do both or did they do nothing? So we're going to add two more chemicals, only to the tubes that didn't turn red the first time, right? Because we know the answer for those tubes. The ones that did not turn red the first time, we get zinc powder. This is the chemical symbol for zinc. Ground up zinc metal powder. And this is hydrochloric acid, HCl. And only the H is uppercase there. HCl. Well, the C is here. The L is lowercase. Right? HCl. Hydrochloric acid. Zinc and hydrochloric acid, those two chemicals together, will chemically convert nitrate into nitrite.
What is denitrification?
Converting nitrite into nitrogen gas and nitrous oxide. That's shift, starting from here and going to there. So zinc and hydrochloric acid, just these two chemicals by themselves with no bacteria involved at all, will chemically convert nitrate into nitrite. So if we add these to the tube, and all of a sudden the tube turns red, it's because these chemicals have converted nitrate into nitrite. And the tube turns red because you already added nitrate A and B, and those chemicals turn red in the presence of this nitrite. So if the chemicals, zinc and hydrochloric acid, converted nitrate into nitrite, did the bacteria do it? No. No. So that tells us that the bacteria is negative for both. Now, I would just caution you on quizzes and tests. I know I put both up here, but I need to know that you know what both is. Okay? They're negative for nitrate respiration and negative for denitrification. So be clear as possible.
So wait a minute, so you said positive for nitrate respiration and positive for D?
No, bacteria are negative for both. The chemicals
Oh, you're on
in
the
it-
first one,
No, I'm on this one.
Negative
Right.
for
on that, yeah.
denitrification and respiration.
Negative for nitrate respiration, right here where I said both. Negative for, right, nitrate respiration and negative for denitrification. The chemicals did it, not the bacteria. Alright, so let's just quickly review. We added nitrate A and B, the tube did not turn red. So we add zinc and hydrochloric acid, now the tube does turn red. That tells us that these chemicals converted nitrate to nitrite and not the bacteria. So the bacteria are negative for both nitrate respiration and denitrification. So if the tube never ever turns red, you've now added all four reagents, nitrate A, nitrate B, zinc, hydrochloric acid, the tube never changes color. That's because the bacteria did both processes. They're positive for nitrate respiration and positive for denitrification. We'll go over that again on Monday when you're actually looking at the results. Today, all you're doing is not really looking at the definitions there. This is on page 83 of your photographic atlas, nitrate respiration lab, page 83.
On, the oxidase, did we have a photo on the atlas?
Yes.
I know you said-
You wanna put that on the website?
I'm going to.
Yeah, because my battery is out.
Okay.
Right there on
Okay,
the website.
so everything you need should be up here on the front table, nitrate